Hi, everybody. Great to be here today. I want to first just start off by thanking the conference organizers, Margie and Danae. I think you've done a phenomenal job, and it's so great to see all three organizations here and all the patients, and the first time that we can be together after a few years of COVID, and I think it's really powerful in moving these treatments forward. So today I'm going to be talking about Pegtibatinase and the initial results from the COMPOSE trial. But before I get started, Margie gave me a great introduction, but actually, what I'm going to be speaking about today and what you may be seeing for the first time actually began a long time ago, more than 10 years ago in Jan Kross's lab. And since then, there have been literally hundreds of people who have worked on this, including many of the people on our team who are here today at Travere, but also so many other researchers, doctors, and the patients and their families who have supported this research and bringing it to this level. So with that, I just want to say I'm incredibly humbled to be here in front of you and really have the honor and privilege to be able to explain where we are with the drug treatment at this time. And I'll stop in between a few slides here if there's any questions, so this can be a back and forth discussion as well. So we always start out with some disclaimers. So the drug that we'll be talking about, Pegtibatinase, it's currently being studied and it's not approved by any health authority. It's being studied for the first time in humans, a first-in-human trial that we'll be talking about, the COMPOSE trial for classical homocystinuria. And the safety and effectiveness of Pegtibatinase are not yet established. And so that's something that we're trying to do through these trials and something that we definitely need all of your help for. I'll just start with a little background on Travere Therapeutics. You may not have heard of us before. We're relatively new to the homocystinuria space. But we are a biotechnology company that's really focused on rare diseases. Travere acquired Orphan Technologies, which is a company that did the initial work, and you may have heard of that before. It was previously called OT58. And so they did a great job really doing the early research and bringing this into human trials. And so now it's up to us at Travere to really take this drug further and really understand, what is the safety and is it an effective treatment for homocystinuria. You can see these are our values. Many of the people in the company are personally affected by rare disease, and that's one of the reasons they choose to work there. We are about 350 members, and we're based out of San Diego, but we have global offices in Switzerland and Ireland. This is just a quick snapshot of the pipeline or the other drugs that the company works on. So we have a drug that's in late phase 3 testing for rare kidney diseases. There's another drug for a rare disease called CTX, classical homocystinuria, and others. And this just goes to show that we as a company are really committed to rare diseases with unmet medical needs. And rare diseases are very different from other types of diseases. And oftentimes it's much more challenging to develop drugs for rare diseases. So you really need a specific type of expertise. So we all know this, and you guys are more familiar than any that new treatments are needed for classical HCU. Now, we talked about in the previous talks about many different forms of homocystinuria. I want to be just clear that today I'm going to be talking specifically about classical homocystinuria. And the reason for that is the drug that we're developing is specific to this disease, and specifically the enzyme that breaks down homocysteine, which has been covered in many of the other lectures, called CBS or cystathionine beta synthase. That causes the lack of activity of the enzyme causes elevated homocysteine in the body, and this leads to many of the complications that occur in patients. The treatment, which has been talked about quite a bit here, low protein diet, which is very difficult to maintain with metabolic formula, as well as betaine, and vitamin B6 and other vitamins. However, even with aggressive treatment, many patients are unable to keep their homocysteine levels below 100 micromolar as recommended, and often continue to suffer some of the complications of homocystinuria. So I'm going to talk a little bit first about our natural history study. And you guys have probably heard a lot about natural history studies. They're trying to recruit over there in the breakout room. So why do we need natural history studies? I mean, can't we just go and do a trial and test a drug? Well, the issue is that rare diseases are often not really well understood, even by the medical community. And you guys are very familiar with this. And so if we don't really understand the disease and what happens to patients, then how will we know whether a drug is improving those symptoms, especially when there's not a lot of patients and patients may have different symptoms that occur at different times. And so that's why a natural history study is really important, especially for rare diseases. And that increases the knowledge we know. It allows us to make objective measurements about those who participate. And those measurements provide a reference point, something that we can really compare when we do have novel treatments and we try them. We can compare them against the natural history and understand whether or not those treatments are actually effective or not. In addition, the natural history really helps us understand that disease and design the clinical trial in such a way that we are capturing the complications or the issues that are most important to patients. So Travere's natural history study began in 2016, and it's been ongoing since then. We are looking at various aspects of classical homocystinuria, including how do homocysteine levels change over time, how are they associated with complications of classical homocystinuria, what are the current treatments being used, and how effective are they? And then very importantly, we also include an impact of diet. Because if we don't add the diet piece to there, it will be hard to really understand and interpret all of the other information we get. And so we hope that understanding these current challenges will help provide us a reference point as we look at clinical trials with pegtibatinase and understand whether or not pegtibatinase could be effective for patients. So this study is ongoing and enrolling. You may have seen some of the flyers at the booth. The goal is to observe patients for over 6 years. And currently we have enrolled 65 patients and we're targeting 150. It's ongoing at 8 clinical sites. I just want to take a moment here to really acknowledge those who have participated in the natural history trial, which some may be in this room. It's really appreciated your participation in providing this information. And we've been asked, when is that information going to be coming out? Well, one, the study is ongoing, so it takes a while. We still need to collect more data. And there are you need several years. It's not information you can do in a few weeks or months. But the good news is that we are analyzing this data and we do expect some of the results from this natural history study, at least the early results, to be available at a scientific conference presented soon. And from there, we'll definitely be communicating with HCU Network America so that you guys will be able to provide the information to you as well. So definitely we'll do that. So what is this natural history study? Well, it's very simple. Anyone with classical homocystinuria who's age 5 to 65 and willing to come to one of the clinics every 6 months is able to participate. And we'll be doing various tests, which are probably familiar to you and would be done in any kind of a visit. And these are all free of charge. And this data will all be collected to really understand what is the current state of the condition. Let me pause here. Any questions? Okay. So let's move on to pegtibatinase. Now, first I want to apologize for this name. We did not pick this name. It was chosen for us, and it's based on the structure and the way that the drug works. And I honestly, I had to practice quite a few times pegtibatinase, just to be able to say it. But what is it? It's an enzyme that breaks down homocysteine. So you guys have seen a lot of pathway diagrams so far today. This is just a simplified one to try to help explain how pegtibatinase works. So as protein is taken in from food sources, it's broken down into the amino acids, of which the primary one that we look at is methionine. And methionine is then converted into homocysteine. And you can see there where the CBS enzyme is, that's where the block occurs. The enzyme is deficient of activity, and therefore homocysteine builds up in the body. With the buildup of homocysteine, this eventually leads to the health problems from classical that we're familiar with in classical HCU. Now, what is pegtibatinase? Well, it's very simple. Pegtibatinase is just a modified version of the same human enzyme, which is deficient in activity in these patients. And so it's been engineered to be a little bit longer acting than the original enzyme, and so that it can be administered under the skin by a subcutaneous injection. Now, we talked a little bit, or we heard a little bit about the drug development pathway, which takes a long time, unfortunately. But there are the different stages of drug development. And of course, before we would ever test a drug in humans, we want to make sure that we know what it does in animals or in mouse models. And we're very lucky that unlike some of the other cobalamin defects that were talked about earlier, the mouse models for classical homocysteinuria actually are very similar to the symptoms that we see in humans. And so mice were created that had the same defect in the same gene, and they actually develop many of these same complications in the eyes, skeleton, brain, and blood vessels. And so a lot of testing was done over the past several years of pegtibatinase in animals. And it was found to reduce both blood and tissue levels of homocysteine, and also was found to be generally safe and well tolerated by these animals. No real safety effects that were found, at least in animals. And when we looked at the complications that occurred in the various organ systems, we found that pegtibatinase could actually improve all of these. And so these are published studies. If you're interested in the science, I definitely encourage you to look them up. So moving on to the clinical trial. So as Margie mentioned, sometimes you can combine phase 1 and phase 2. Oftentimes phase 1 is done in healthy volunteers with small doses, trying to understand how does a dose relate to a drug level and is it safe. In this case, with an enzyme replacement therapy, sometimes it may not be safe to give that to healthy volunteers. And so some drugs are actually started, especially in rare disease, are often started as a phase 1-2 that goes directly into the patients who are affected. And so this is a study that was began. And of course, the first priority for any drug that's being tested is safety. We want to ensure that it's safe for patients, and specifically in this trial, we're looking at adults and children with classical HCU who are age 12 or over. Then we want to understand, how does pegtibatinase behave in the human body? How does it affect homocysteine levels? And then when we look long term, are there effects of pegtibatinase on the complications of classical homocysteinuria? And that's really where you'll see that the natural history study would be so helpful. So to understand what complications would occur, so that when we look at this trial and look at the data long term, we'll be able to understand if there's a benefit from the treatment. Any questions so far? All right, so we'll get into the trial now and some of the results. Just starting off with the study design. So this is a little bit of a complicated slide, but really it's telling you how the trial is structured in 3 main pieces. The first is the screening period, where we determine if a participant is eligible. The second is the main treatment period, which is a minimum of 12 weeks. And at the end of the main treatment period, there's this optional treatment period where all patients can continue to be treated with the drug if they would like to participate. I'm going to focus in on this main treatment period here. So the way this study was designed, which is very common in early stage kind of clinical trials, it's a dose escalation trial, which means there are different groups of participants and each of them get a higher dose. And you start with the lower dose, you see if it's safe and tolerated, and then you move up to the higher doses. Importantly, for each of these groups, there were 4 participants, and those participants were what we called, it was randomized double blind. So that means that one patient out of the 4 received a placebo or a injection with no medicine. The other 3 received the medicine of that dose of that group. And none of the patients and none of the doctors knew who was getting the treatment and who was getting the placebo. And it was randomly, the treatment was randomly assigned. Now, this is really important, this randomized placebo control, because homocysteine levels can vary pretty significantly, especially with diet and other factors. And so if you don't have that placebo control, sometimes it's hard to provide evidence that any changes in homocysteine are really due to the drug. And so that's why this design was selected. And so we had 4 patients in each of the cohorts, except for group 4, where we only had 3 patients. That was related to some challenges with COVID. But overall, a total of 19 subjects who were enrolled in the study. And of these 19, 18 completed the 12-week period. 1 participant had to stop the trial due to unrelated reasons. Any questions before I move to the...
We have 10 minutes. You might not want to take questions.
Okay. Okay, you got it. So just briefly talking about who were these patients. So there was a total of 19. Of those, 4 were pediatric. You can see that the majority of patients were on betaine and vitamin B6. About half were on formula. Their starting homocysteine levels were pretty high, between 130 and 180. They were mostly male. The average age was 24 when the study began, and race, they were mostly white. Let's talk about the initial safety results. I'm pleased to say that across all 5 doses tested, Pegtibatinase was generally well tolerated. The side effects that were seen were mild and did not last long. And importantly, they did not get worse as you give a higher dose. The most common side effects were injection site reactions, injection site redness, injection site pain, things that are somewhat expected given that this is a drug that's given by subcutaneous injection once or twice a week. We also had 3 patients develop hives or a little bit of swelling around that area, but these were mild and resolved. We look very carefully at something called serious side effects, and there are criteria that determine that. And there was only 1 serious side effect that was reported that was considered likely related to the drug. It was a case of acute hives, and the medication was stopped for 1 week. The hives cleared up after 11 days. The patient was able to restart the medication and had not had any recurrence of the hives, so that was good news. Overall, no patient stopped the drug due to any side effect related to the drug. Importantly, there were no severe allergic reactions or immune reactions in that respect, and that's one of the main concerns when you think about enzyme replacement therapy and other drugs in this kind of category. We're always looking for immune reactions, so we did not see that. And otherwise, the general blood work and other testing we did was unremarkable. So now we're going to look at the results, the initial results, and try to understand how does Pegtibatinase affect homocysteine levels. So in this graph, you see this horizontal line. Anything above the line is considered increased homocysteine. Anything below the line is decreased homocysteine. And what we're doing here is we're looking at each subject in each cohort and understanding how did homocysteine change from the time that they started, or what we call change from baseline. And the different bars and the different colors indicate the different groups. So the first group is placebo. The next one is group 1, which is the lowest dose, all the way up to the highest dose in orange, which is 1.5 milligrams per kilogram twice weekly. So what you'll see is as we increase the dose of pegtibatinase, we get a larger and larger decrease in homocysteine, which is particularly noticeable in groups 4 and groups 5. Now, group 5 had the largest decrease of 55%, an average of 55% at the end of 12 weeks. Now, there is some fluctuation in homocysteine values. So we do know that you may see your 5 or 10%, et cetera, change. But as we increase the doses, we're seeing that directional movement. Now, I want to focus in a little bit more on the patients who received the high dose and try to understand how did their homocysteine levels change as compared to those who got placebo or injections with no medicine. So there are two graphs here. The left one showing homocysteine and the right one showing methionine. And we talked a lot about how methionine is a really important marker in classical homocystinuria. And so it's important to monitor that as well. So starting with the graph on the left, we have homocysteine on the y-axis, and across we're looking at time over 12 weeks. In the gray line, we have the placebo. And you can see they started around 130. And over that 12 weeks, there was some fluctuation. But for the most part, there really wasn't much change. When you look at the high dose group, pegtibatinase 1.5 milligrams per kilogram twice per week, you saw that they actually started at a much higher level, around 180. But within 2 weeks, they had a rapid decrease in their homocysteine levels. And this average homocysteine level actually came below 100 and was maintained below 100 for the entire 12 weeks. When we look at methionine, we see an even greater effect. So again, the pegtibatinase group started off higher, but within 2 weeks, 3 weeks, very rapid reduction and an average of 82% reduction. So overall, we find this data very encouraging for the ability of pegtibatinase to be able to lower homocysteine and keep it reduced for a long period of time. So how might we put this information all together and understand? How might this actually affect the disease and the homocysteine and methionine levels in the body? Well, pegtibatinase, when given under the skin, it functions in the blood. But we know that this conversion of methionine to homocysteine occurs in the cells. So what we think that happens is that as you put pegtibatinase into the bloodstream, you get a breakdown of homocysteine and a reduction. And this leads to a reduction and a movement of homocysteine from the cells into the plasma. As you reduce homocysteine in the cells, you get a reduction of methionine, and this ultimately leads to what we saw in the clinical trial, which is a reduction of methionine in the blood vessel. And so we've come up with the idea of calling this a metabolic sink, because when you have a blockage in CBS, you basically get a backup, just as if your kitchen sink was blocked up and the drains clogged. However, once you add pegtibatinase to the mix, you're able to break down homocysteine, and this suddenly releases the pressure and allows you to reduce homocysteine methionine in the cells. And that's something that we've confirmed in our animal studies, and ultimately leads to reduction of methionine as well in the blood vessels. So just to conclude the results, pegtibatinase was generally well tolerated at all the doses tested, and there were no reports of severe immune allergic reactions. No patients stopped the study drug due to side effects. Patients on the higher doses had this rapid reduction, and the patients with the highest dose had a reduction of about 55% at 12 weeks. In addition, this reduction was sustained below 100, the level of 100, which is really critical from a clinical standpoint. And overall, these are initial results from this trial. This trial is continuing to be ongoing, and much more study has to be done. But these initial results suggest that pegtibatinase may have the potential to be a new treatment for classical homocystinuria. Now, our work is not done yet. This is just an initial look. We're still enrolling another group of patients at a higher dose, and we very much would like for people to participate in this study to allow us to find out which dose is going to be the best dose. The next step for drug development is to get to a phase 3 study. And a phase 3 study is a much larger version of what we're doing here. And it's just much bigger in general. And it's to confirm the effect, the safety and the effect in a larger group of people. And that's something that, before we proceed into we have to get agreement with the FDA and other regulatory authorities. So we're working hard now, talking to the FDA and trying to get an agreement. And so far, things are looking good. And then we're also working closely with doctors and patients and patient groups to really understand, what is it in classical homocystinuria? What are the issues? What are the concerns? And what is the potential role of pegtibatinase treatment? I apologize that my acknowledgement slide somehow came out. It didn't come in here. But just I want to end by giving my real heartfelt gratitude to the patients who have participated in this trial. It's very difficult to participate. I mean, it's inconvenient. And these patients have really made a sacrifice to really try to help us move forward. So the patients who participate in COMPOSE or the natural history study, or actually any trial, honestly, any trial, any registry, I really, really encourage you to participate. There is no way to develop a new treatment if patients don't participate and really get active with research that's ongoing. So with that, I'll close. Thank you very much.
Take one question and then the rest.
Sure.
Bring in another panelist.
I had a question with regards to, well, it's great to see that the methionine, the homocysteine levels on the cohort 5 have been reduced. And you may or may not be able to explain this to me at this time because this is layman asking a question.
No problem.
However, what I was wondering is with the reduction in the enzyme that infiltrates and moves through the body to reduce, while we collect you collect laboratory blood draws periodically, I'm sure with the research on designated time frames. So my question is, with that enzyme injection, whatever the schedule is, in between, is that job that the enzyme is performing immediate and not sustained between injections, or does it somehow stay within the system?
Sure.
Do you know that, or?
Sure. Yeah. So when we start these studies, we do what's called pharmacokinetic sampling or PK sampling, which means that we try to detect how the levels of the drug go up and go down over time. And so we do that testing in COMPOSE early on to really understand. So definitely when you first give the drug, that's when it's most active. And over time, the effect goes down. And so what we do is we check all these results we provide are what's called a trough, or at the very lowest point. So when you give the drug, the doses go up, homocysteine levels come down, but we don't want to know what happened just right then. We want to know what happens before we give the next dose, because that's when the drug would be at the lowest concentration. That's when homocysteine levels might be at their highest level. But we do test them over time and in between in between doses as well.